So we'll move straight on to another hot topic, and we've had quite a few presentations today thinking about CMV and thinking about the CMV infection of the patient. We're moving over to thinking about CMV, the virus, and I'm delighted that we've been able to be joined by Jocelyn Hume, who is one of our scientist colleagues. Jocelyn is in the final year of her PhD, and she's got a really great project going and a great interest in CMV and antiviral resistance, and has been doing fantastic work in this, and is looking at the burden of CMV in transplant patients throughout Queensland, specifically on resistance. And so I'm really looking forward to seeing what you're going to tell us about the virus, because we've heard so much about the host. Thanks, Jocelyn.
Thanks, everyone. So I'll just firstly mention my supervisors, Emma, David, Kim, and Adam, and thank them for their ongoing support. So we have received a grant from the Children's Hospital Foundation to investigate stem cell transplant, CMV infections in stem cell transplant recipients. So I'll just give a bit of a background about CMV. So it's a type of herpes virus. It has a seroprevalence of approximately 40% in infants, and that increases to about 80% in adults. And you can see the modes of transmission on the right here. In healthy individuals, CMV causes a mild illness, but like all herpes viruses, it causes a lifelong latent infection. So in immunocompromised people, like transplant recipients, CMV can reactivate, and it can infect almost any organ in the body, causing severe disease and increasing the risk of mortality. So CMV replicates in many different types of cells, connective tissue, epithelial cells, neuronal cells, but it becomes latent in the progenitor cells, and it hitches a ride on monocytes around the body to infect other cells. In transplant recipients, CMV, it can be transferred in the transplanted organ, or it can be reactivation from a previous infection. Solid organ transplant recipients, they're at higher risk from donor seropositivity, but stem cell transplant recipients are at higher risk from their own seropositivity. So early CMV reactivation in these patients is often asymptomatic, but when they do get symptoms, it can be very similar to rejection, graft versus host disease, or medication side effects. So the symptoms is really hard to judge based off of the symptoms. So that's where testing comes in. And to make it even more challenging, clinicians need to perform a delicate balancing act between the amount of immunosuppression to prevent graft rejection, but then not so much that it causes CMV reactivation. So that brings me into testing for CMV. At Pathology Queensland, we perform CMV PCR, which is a qualitative test, so it tells you whether CMV is present, not present, but it can tell you, based on the specimen site, potential organ disease. So, for example, CMV detected in vitreous fluid could be indicative of CMV retinitis. At Pathology Queensland, we also perform CMV quantitation, which measures the viral load. And guidelines, as Brad mentioned in his talk earlier, the guidelines aren't really clear, but for high-risk patients, they're usually monitored for weekly for their viral load for about 100 days post-transplantation. So thankfully, we do have quite a few drugs to treat CMV. First line, ganciclovir, or its orally available valganciclovir. Second line treatments, foscarnet and cidofovir, both of which are administered intravenously. And then two newer drugs, maribavir and letermovir, which have been recently approved by the TGA in the past few years. And all of these drugs have toxic side effects. So obviously we don't want to give drugs unnecessarily to give these side effects. So there are 6 genes involved in CMV antiviral resistance, and each of these genes encode a protein, which has a different role. They have different roles regarding CMV replication. And the drugs that target these genes, mutations in these genes cause resistance to these drugs. For example, the UL97 gene encodes a viral protein kinase, which is required to phosphorylate ganciclovir so that it can act on CMV. But mutations in this gene can cause it to not work on ganciclovir, hence ganciclovir resistance. So I hope you're all paying attention because there's going to be a quiz. Kidding. This slide really just shows that there are hundreds of mutations that cause CMV antiviral resistance. And in this box here, you can see that there's quite a few mutations that cause multi-drug resistance as well. So this is kind of highlighting the importance of actually detecting which mutation is causing resistance. For example, if a patient was being treated with ganciclovir and developed one of these mutations, then they would also be resistant to foscarnet. So it's important to know that information beforehand to help treat the patient. So let's talk about the current testing for resistance. Currently, there aren't any local testing for CMV resistance here in Queensland. Instead, samples are sent to Sydney, where they perform Sanger sequencing. Sanger sequencing isn't very sensitive, and it's not able to detect minority variants of less than 20%. And the Sydney lab only tests for mutations in the UL54 and UL97 genes. So if we go back to our previous slide, that means that all of these mutations highlighted in red can't be detected. So no letermovir resistance is able to be detected currently. The turnaround time as well is not great. It's up to about 3 weeks, at which point the resistance results aren't that useful to us. So there is a need to improve CMV antiviral resistance testing. So we wanted to address this by looking at next generation sequencing assays. The reason being that it has quite a few advantages over Sanger sequencing. For example, it's able to measure the variant frequency. So it can tell you whether a mutation is 20%, 50%, 100% the population compared to the wild type. So that can mean some differences in the clinical significance of that mutation. It can also detect minority variants of less than 20%, unlike Sanger. And this citation that I've just put at the bottom here is just one example that talks about the importance of minority variants. Next generation sequencing also has other advantages over Sanger. For example, higher throughput, meaning that you can sequence multiple genes at the same time. So that also means faster turnaround times. So we had a look at 2 commercial research use only assays, the Vila Diagnostics assay and the Agilent community design CMV assay. And we also designed our own amplicon assay using the Illumina microbial amplicon prep kit. So let's look at these side by side. So the Vila Diagnostics assay and our in-house assay are amplicon assays. So that means that they are only targeting the genes involved in resistance, whereas Agilent whole genome sequencing, so it's obviously sequencing the entire genome. The cost for these, both the commercial assays are quite similar in price, whereas our in-house assay, as expected, was quite a bit cheaper. Now, these costs are just for the consumables and reagents, so please don't get upset later on if pathology says, oh, it's a lot more than that. It doesn't account for person hours. So the turnaround times for these, both the commercial assays, they require 2 days of library preparation. So you add on an extra day for sequencing, so it's 3 days turnaround time. Our in-house assay only required 1 day of library prep, and it was super fast, super easy. So add on an extra day for sequencing, that's 2 days turnaround time. Sorry, my boxes aren't really lining up here. So for the the threshold that the companies state. Vila Diagnostics state that they have a lower threshold of 1,000 international units per mil. That's pretty much on par with what Sanger sequencing requires. We were able to detect resistance in a sample lower than that, but we also had quite a few failures above that. For their adjuvant assay, it states 11,000 international units per mil. So that's actually, because it is whole genome sequencing, they're looking at getting good coverage of the entire genome. But when we looked at just the 6 resistance genes, we were able to get pretty good coverage for those, down to about 2,000 international units per mil. And for our in-house assay, we haven't stated a threshold there, but we got pretty good coverage down to about the same levels. For automation, the Vila Diagnostics assay is semi-automated, and the adjuvant assay, it can be automated. They do have an instrument for the automation, but it does also come along with extra cost. And our in-house assay, there could be the potential of automating that using third-party equipment. For the analysis, the Vila Diagnostics platform, it has the analysis on board, which can be really, really useful, and it's got really clear reports that it does spit out. Unfortunately, the adjuvant and our in-house assay do require the user to perform that analysis. A few other things to note with these assays, the Vila Diagnostics assay, it can be mixed and matched with hepatitis C, hepatitis B, and HIV typing. So that can be really useful. You can batch up to about 15 samples at a time. So that can be really useful for laboratories with small sample numbers wanting to look at multiple infections. Unfortunately, we did have a few problems with the Vila Diagnostics assay. Poor amplicon design, which I'm going to come back to in the next slide. A few database problems. So the database was out of date, it was missing quite a few mutations, and it was also including mutations that weren't clinically significant. So it was overcalling resistant. So that's a pretty big problem. We also experienced numerous technical issues throughout as well. With the Agilent platform, because it is whole genome sequencing, you're looking at 236,000 base pairs, when really, if you're looking for resistance, you're only interested in about 20,000. So you've got a lot of extra data there that may not be necessary. But it does have the positive that if you are interested in a different area of CMV, or you want to look at genotyping, it can be very useful there. The Agilent platform as well, it can be made compatible with Illumina, 454 pyrosequencing, or ion torrent sequencing. So it can fit into existing sequencing facilities without the huge upfront cost of implementing that. Our in-house assay, it's easily customizable. So if we wanted to add in a gene for genotyping, we can do that. If a region is dropping out, not sequencing quite as well, we can add some other things in there to boost that. So that was a big positive with that. So what I was saying earlier with the amplicon coverage with the Vila Diagnostics assay. So here I've got the 6 genes that are involved in resistance, and the green highlighted areas are the amplicons covered by the Vila Diagnostics platform. So you can see that there are 2 mutations outside of that region that cannot be detected with this platform. So that kind of means that kind of needs like full redesign to be able to detect those mutations. For our amplicon assay, we've actually designed it to cover the entirety of all of these genes so that, if new mutations pop up in a random spot, we can detect those without redesign. So you want to hear about some results. Here I've just got the 2 genes because 6 was too many to put on a slide. So the UL54 and the UL97 gene, and I've just plotted the trend lines for the coverage for all of the samples that I ran on these assays. And as you can see, as the viral load increases, the coverage also increases as expected. The blue line, Vela Diagnostics, it obviously caps out around the 60%. That's because of the amplicon design not covering the entire genome. So overall, in all 6 genes, the Agilent platform appeared to perform the best overall. But all of these assays have the potential of detecting CMV antiviral resistance, and all of them have their own pros and cons. So laboratories looking at implementing these assays need to pick the best assay for them. Other work that I'm doing, I'm developing a bioinformatic pipeline to detect CMV resistance so that we can actually have a look at the mutations that we have looked at for the Agilent platform and for our in-house assay. I've also designed a prototype CMV assay, viability assay, to differentiate between live and dead CMV. And we've recently had ethics approval to collect patient clinical data. I know Julia really wanted me to talk about it today, but I haven't quite gotten there, so I'm so sorry. Please watch this space. Hopefully in my next talk, I'll be able to talk about it. So just to summarize, CMV antiviral resistance is a significant burden for transplant recipients. Current testing for CMV antiviral resistance is insufficient. To address this, we've had a look at 3 next-generation sequencing assays, and all of these assays have the potential for detecting CMV resistance. We're also developing a bioinformatic pipeline to analyze these. And we hope that the implementation of one of these assays will improve the diagnosis of CMV antiviral resistance and ultimately improve outcomes for transplant recipients. So thank you so much.
Thanks, Jocelyn. Amazing tour de force run through of many years of work, I think. Thank you.